applications that go as follow, and is connected to the fear or to the concern about the fate of organisms that we manipulate in the laboratory when they are released deliberately or accidentally. So there are some questions here born by that are logical and legitimate to raise for laboratory designed microorganisms, whether they are GMOs or whether they are synthetic bioagents. For instance, can they colonize and take over natural microbial communities? Okay. The answer at this day is no, and it's not just an opinion, it's just in every massive expedition that has been going on to say, catalog the environmental microbiome of the whole planet, never ever a hint of recombinant DNA has been found, meaning that probably there have been leaks and recombinant DNA have been out there, but it has never prospered too much, okay? So, but this is a legitimate question. Can, say, recombinant bacteria, recombinant organisms take over natural microbial communities? Is there any chance that they enter niches that natural bacteria cannot? Interesting question. Might they go into a stage of uncontrolled growth? Imagine, I don't know, that we develop in the laboratory a bacterium able to eat plastics. If really is that, if it is released into the environment, will that destroy our entire civilization? I don't know. We can discuss that. Can designed organisms trigger allergies to other undesirable immune reactions? This is a very important question. What are the chances of horizontal transfer of synthetic genes to novel recipients? Another legitimate question. Is there a trade-off between safety and biotechnological efficacy of designed organisms? We say, well, sometimes we enter many, say, features, we will discuss in a moment some of them, to ensure that recombinant bacteria are contained in a given place, but then the downside of that is that they are less fit, and therefore, in many cases, they are not good enough for biotechnological applications. Now, can we engineer traits that evolve towards virulence? Interesting question. Are there scenarios where these bacteria or these organisms can damage life or property? To this day, that has never ever happened, but it's a question to re-say. What is the environmental fate of synthetic genes? Now, number 10 is the real, say, good one. Are there chances of malicious misuse? This is something that I would like to, on Saturday, as I anticipated, we'll have a seminar on these things. You are welcome to join, or not, as you wish, but we will deal exactly with these societal consequences of synthetic biology. And should these bacteria or these agents be endowed with traits to increase their safety and predictability? So that means that in general there is a kind of concern to always increase the predictability of bacteria or organisms that you develop in the laboratory in order to, be safe and avoid possible risk and so. So different laboratories around the years have developed different propositions on how to increase predictability. And some of them are very, say logical. For instance, in the chromosomes of bacteria, there are mobile elements, that generate genomic instability. One way to avoid it is to edit the genome to delete these mobile elements. We can also recode translation, as we discussed in previous days. We can ensure that the gene of interest that we are putting inside the cell overlaps with an essential gene in such a way that the gene of interest cannot be lost because it's intermingled with an essential gene. That's a good approach. We can have a kind of repository of the population that makes the job, and once in a while we refill the target with that population. We can also play with the specificity of transcriptional factors to ensure that if we have a device where we have something expressed under the control of a transcriptional factor, then the system goes on. But, yes?
So when you showed that gene overlapping, so they are the sense and the antisense?
Okay, well, this is a very, thank you for the question, because that enables me to explain this important thing. So when you have a gene of interest in a biotechnological setting. Unless you have a tremendous pressure, for instance, with something, then the gene may be lost because the cells normally don't want to be implanted with something that comes from the outside. So how can we secure, how can we ensure that a construct remains durable in a cell without any external selection? Okay? This is not an easy question, okay? So there are different ways of, different propositions. And the standard proposition is to engineer some circuits, and in a subsequent class I will discuss some of them, to kill the cells that misdo what we have programmed them to do, okay? But that's a negative selection. So other people have thought differently. Say, well, what if what we do is that through bioinformatic tools, we intermingle the sequences of the gene of interest with some other sequences that appear in essential genes. So if you do that, what happens is that if you, whatever reason, you have a mutation here in this gene of interest, then the mutation will also affect an essential gene, and therefore the cells will not grow. Therefore, it's a way of selecting for maintenance of something that we engineer in the genome. Obviously, this requires a massive computational power because you have to find sequences of homology between one gene of interest and so on. So, but there are some applications that conceptually are very bright, playing with this idea of gene overlapping, gene intermingle between an essential gene and the gene of interest. Okay? Good. Now, how can we increase the predictability by other means? Well, the predictability by other means, or mostly to avoid the undesirable consequences of this instability, it could be entertained in terms of containment. So containment means that we want this particular construct to work only in time and space in a predefined setting, and make sure that it will not escape that particular setting, okay? So there are various approaches to that. Well, you may have a physical containment. I mean, you make a laboratory with all types of barriers in such a way that your constructs never escape it. Or you can have a system where you kill the DNA and then you have a cell-free system. And then that system cannot proliferate. It can make its job during a period of time, but it will not proliferate because it doesn't have DNA. But you can have other, say, approaches, right? So one very, very, well, I want to say old, because it was developed in the 70s, but it's a kind of trivial one, is to make your strains auxotrophic for something that they need to grow, okay? So that means that if, for instance, your strain is auxotrophic for, I don't know, serine, then only in places where you have serine available, the strains will grow. If you don't have serine, they will not grow. By the way, did you see the movie Jurassic Park? Yes. Okay, you remember what the dinosaurs were auxotrophic for? I don't know if you remember the story, or maybe you were too young to understand the story. But that movie has this detail. They say, well, we have these dinosaurs in an island, and then this island, but we want them to escape. We don't want them to escape, right? So to make sure that they never reach the island, we engineer them to be auxotrophic for, I think it was for methionine, okay? And then in the food that we give them methionine. But then one of them escapes because it finds a tree somewhere that has a lot of methionine, and then the dinosaur is able to propagate around because he loses. So in fact, this movie was made after the proposition of having auxotrophy as a way of containment. So there's some kind of scientific angle in that movie. Okay. Okay, then you have inducible expression systems. You can have safety circuits, like punishing the ones that escape a given time and a given location. Then the last tendency is about this synthetic auxotrophy, that is to make the bacteria or the microbes or the biological system auxotrophic not for a natural compound, but for a synthetic compound, okay? And in such a way, you have all the, I mean, in theory, you have full control of the growth of that particular genetic, that particular microbe, because only if you provide a synthetic compound, then the bug or the microbe will grow. or you can have compartmentalization, as we discussed in previous days. Okay. There are many other propositions that have to do with having orthogonal models, semantic firewalls. You will see in a moment is about changing the genetic codes in such a way that if your engineer organism escapes a given place, then it exchanges genetic materials with others. The genetic materials will have a different code and they will not understand each other. By the same token, genetic firewalls to inhibit horizontal gene transfer, metabolic firewalls. So you can have a whole of, say, possibilities to contain your specific bacterium in a given place. And one of the most conceptually appealing ways of this containment is about this what I call semantic containment. So semantic containment means that because you have a different code in your DNA and in your nucleic acids, if your recombinant DNA escapes into other hosts, then it could not be understood. So because only when you have the right ribosomes able to decode the new genetic code, then the proteins will be produced, right? So remember that yesterday we discussed that the technology today enables to change massively the codons that you have in a place, in a sequence, and therefore you can reassign codons or delete codons or put codons and even have quadruplets instead of triplets. And therefore, in these cases, you get a genetic code that is not understandable by the natural, say, organisms out there, and therefore you have a degree of containment. Okay, so these are conceptually very nice ideas, but again, Jurassic Park, life always finds a way, okay? So that means that regardless of how smart we are, how many semantic containments we are, how much containment, physical containment we have, and everything at the end, some of these guys will escape, okay? And when they escape, they can start proliferating, and then the containment is of no use, okay? Okay, so what can we do when you have this situation, okay? So should we just say, okay, then forget about synthetic biology, and then we go home and do other things? No, no, I mean, there's always a way to rethink on the possibilities. And in this case, the way to go, or at least the way that we have proposed to go, because we are very active in this particular thing, is to change the focus from containment to traceability, okay? So given that the things that you put in the environment will, that you use in the laboratory, sooner or later they will escape, okay, sooner or later. Then instead of crying about that, then let's celebrate it, okay? And then what we have to do is to develop technologies that enable us to follow the fate of what we put in the environment, giving up the possibilities of containment, because as I said, containment may not work eventually. So we have gotten inspiration from this situation. I don't know how it works in India, but in many countries, if you have a pet in your place, you have to microchip it. You have to inject a chip, and then the chip will tell you, take you immediately to a database where you have all the origin of the pet, whether they are vaccinated, whether it is dangerous, whether it has had some diseases. And also, if you abandon your pet, then you are liable legally. You cannot abandon your dog in the street if you have previously adopted it, okay? So by the same token, so you can think on barcoding the DNA of bacteria that are going to be released into the environment, or bacteria that will be used in biotechnology with a DNA barcode. So obviously the DNA barcode requires some specific, say, characteristics. So this is a concept that comes from this gentleman here called Natalio Krasnogor that works in the University of Newcastle in the UK, and we have collaborated with him a lot. Okay, so the idea here is the following. What if we find in the entire, say, landscape of possible DNA sequences, some sequences that have never been biological and can be, in fact, certified not to be biological at all, and we use them as a, say, barcode to put it in the chromosome of the bacteria that we're interested to handle, and then we associate the barcode to a database where we can immediately identify many of the properties that you have in this bacterium. For instance, what species it is, what strain it is, who is the owner, and that's important, both from the point of view of keeping the intellectual property, but also from the point of view of the liability, because by the same token that you have this chip in the pet, and if your dog does something wrong, you are liable. By having this barcode in the genome of your bacterium, if the bacterium does something wrong, you are liable as well. Then you can go to the phenotype. You can, in this database, you have what are the antibiotic resistances, what is the pedigree, what are the safety aspects, and what countermeasures should be taken in case things go wrong. Okay, so what we did was to build a kind of pipeline to barcode bacteria for environmental and or industrial applications. Let me tell you at this point that obviously the barcodes are applicable not only to bacteria, but to all types of biological systems, all type of, say, different, organisms, not only bacteria, but also yeasts, plants, mice, you name it. So here is the computational part of it. So the computational part of it is the challenge of identifying barcodes, genomic barcodes that are about 150 base pairs that have no equivalence at all in any of the known, say, universe of biological sequences. Okay, so that means that you have to go to a database and make subtractions, and it's complicated. But at the end, you have a large number of barcodes that you can commit. You have it in a database, and you say, well, I want to tag this particular bacterium with a tag. Then you ask the system to give you a barcode, and then the barcode obviously is an DNA sequence, right? Then the barcode is generated by a computer, and then by using some genetic techniques that I will not discuss here, even though I will mention them much later, can be inserted into a permissive region of the genome of the bacterium or the biological system that you want to handle here. Okay, so that's something that you get the DNA sequence, and at the end you use that DNA sequence that is an unique identifier. Then you put it in a permissive site of the brain. Well, sometimes the kind of wet part of the insertion is a bit complicated, because to make the system universal, we had to develop a way of inserting DNA into the genome of the target biological system that does not depend on recombination. This is something that there's a lot of genetics here. I will not get into that, but just to tell you that we have and developed, and others have developed, platforms that enable entering DNA segments of that size in all types of biological targets. And in this particular case, a technology that is called target-on technology. I insist that I will not discuss this today, but the technology exists, and basically with this technology, you can tag, you can add unique identifiers to every type of biological system. Well, this is just to indicate that these barcodes are introduced in very conserved, but permissive sites of the bacterial genome. There's one in particular that is close to the TN7 insertion site. Well, these are details, I don't want to get into it, but technically it is possible to do it. Now, once you have your strain barcoded, what do? You want to identify it. Well, what you do is that you get a sample. The sample can be with an isolate, or it can be in an environmental, say, sample directly. You PCR up the region of the where the barcode is, and you sequence it. And then the sequence will take you immediately to a digital twin. Digital twin means that you have a database with everything that is known about that particular bacterium. And therefore, for you, it should be easy to identify, not only to identify the bacterium with all the different features about intellectual property and all these things, but also to have a scientific insight on the physiology and the lifestyle of that particular bacterium. So we can discuss or not whether we have to still bet for containment or betting for traceability. Maybe the two at the end are good tools to descend, even though we have to keep in mind, as I mentioned before, that perhaps the certainty of containment, 100% secure containment, is an impossible endeavor, okay? If some of you have a good idea on how to handle that, you will become multimillionaire, right? So maybe one of the IM projects that we will discuss the last few days, you have a good idea on how to handle, how to increase the certainty of containment or the say complete containment. Okay, so so far we have been discussing chassis in general, you know, all the properties, containment or not containment, barcoding, what is a chassis, what is not a chassis, the nomenclature, etc. But in this part of the talk, and I would like to spend giving you a specific example on how a natural isolate taken from soil ends up being a super strong synthetic biology chassis. Okay, so as I have told you, and if not, I tell you now, in my laboratory we are interested in environmental scenarios. So the question that we have been studying for a long time or interested for a long time is to what degree we can use genetic engineering and synthetic biology to generate agents, to generate bacteria able to act in this type of scenarios as bioremediation agents, okay? So, and the starting point is that these places used to be colonized by bacteria that, you know, you don't see with your eyes, but when you have the right microscope, you see that these places that are smelly and terrible, when you look at them, they are rich in life, and this life is composed for the most part of microorganisms that have developed the ability to use the chemicals that are here as carbon and energy sources. And therefore that means that they have genes that we are very willing to take to the laboratory and to kind of train and domesticate and use for our own purposes, so that at the end we have something that we can put back into this type of scenarios to make a good job. So many of these, well, there are, you know, thousands of different isolates of bacteria that grow on interesting chemical compounds. So here I just put a very, very small fraction of bacteria that grow not on glucose or fructose that are very boring, substrates, but in very complex molecules. So these are fast growers. So that means that you go to a place, you enrich the plates, you take a enrichment culture for bacteria that are able to grow fast in given, say, compounds, and then these are the ones that you isolate. And if you do that, you are likely to isolate many bacteria of this type. For the most part, gram-negative bacteria, and many of them belong to the genus called Pseudomonas. So I have some of them here, and, you know, I've shown this slide thousands of times, but when I see the compounds that they're able to degrade, I cannot but be amazed that they are able to grow happily on compounds that for us are environmental pollutants, okay? So we have some bacteria that grow on toluene or xylene, others that grow on phenol, that is really bad, phenanthrene, 3-methylbenzene, naphthalene, aryl esters, 2,4-D, a horrible compound, 3-chlorobenzene, horrible compound, aniline, alkenes, you name it. And in the top, we have this interesting bacterium called Pseudomonas putida MT2 that was isolated time ago on the basis of its ability to grow on toluene and M-xylene as the only carbon sources. When you have a spill of toluene and M-xylene, that's the end of life in the ecosystem affected. However, this bacterium manages to use these compounds as gourmet food. So this bacterium is so happy when it finds these two compounds in the environment. Okay, so so that's the starting point, and therefore what I'm going to tell you is how this environmental isolate that was taken from this mixture of different and nasty bacteria ends up being a super nice and super predictable chassis. So Pseudomonas in that respect is just keep in mind that because of the place where it has been isolated, the bacterium itself is already adapted to perform under very, very harsh environmental conditions. And that's very good for industrial biotechnology, and it's very good also for environmental biotechnology, because you are already used to live in nasty places. So if you improve this bacterium to do something new or interesting, it's so much better because they are used already to live there. Okay, so this is the habitat of P. butida, very bad places. But once this bacterium is isolated, then the first thing that you take a look is to its genome. Okay, and well, you know that now sequencing the DNA of genomes is very simple, and so the genome of P. butida was published in the year 2003, and still to this day is a source of very important information. So what is interesting in this bacterium? Well, you know, and I think that some of you work in this field, that out of the genome of some bacteria you can immediately generate a metabolic model. And in the case of Pseudomonas, there are various models here that have been improved with the time. And well, when you see what is going on in these metabolic networks, you immediately recognize something that is very interesting. And the main feature of it is that the metabolic network seems to be kind of evolved not so much to generate biomass, but to tolerate stress. So that means that the entire genome of, entire metabolic network of Pseudomonas butida seems to be very interested to produce large amounts of reductive power, NADH and NADPH. So the interesting part of Pseudomonas butida is that it has a good, what you call a metabolic heart. And a metabolic heart, you know, our mechanical heart pumps blood in all our body. The metabolic heart of Pseudomonas butida produces a lot of NADH, NADPH. If you are interested, you can take a look to the different papers, but you know, just take my word that the metabolic network of Pseudomonas butida is is kind of predisposed to generate large amounts of NADH and NADPH, what makes this system very good for precisely this type of environmental and industrial applications. So when you examine then the genome, what do you get out of it? Okay, this is something just by looking at the genome, okay? You don't have to do anything else. And this is something that you can do these days with different types of bacteria. Well, one thing is that it's a bacterium that is solvent tolerant. So that means that it's able, as we discussed before with Jarrovia, to work under conditions of apolarity, and therefore, well, for some industrial and environmental applications, is not a bad thing. Then the second aspect is that, as I just mentioned, the metabolism generates large amounts of NADH and NADPH, and this is very good for hosting reactions that are redox, serious redox reactions. So that's quite good. Then it has a diverse metabolism, so that means that it can grow not only on glucose, fructose, and these things, but you know, it can grow on toluene, xylene, different aromatics and different compounds that are not usual and that are often found as components of environmental pollutants. And also, and this is important, it's a safe host of recombinant DNA. Let me tell you at this point that Pseudomonas, the Pseudomonas genus is like a family with good guys and bad guys, okay? So the bad guys are Pseudomonas aeruginosa, okay? And maybe some of you have had, well, maybe all of us have had at some point an infection of Pseudomonas aeruginosa in the ear or something. They are really nasty. And sometimes Pseudomonas aeruginosa can create still more havoc by infecting the lungs of patients with cystic fibrosis. And so we have a branch of Pseudomonas that are the bad guys. Pseudomonas butida belongs to the good part of the family, so that means that they do only nice things. They smell bad, I have to say, because the name putida means that they smell bad. However, they are wonderful bacteria otherwise. But being wonderful does not avoid them to have also some bad traits. One of them is that the strain itself is highly resistant to antibiotics, to different antibiotics. It has a complex cell surface. That is something that we may not like for some purposes. It has many prophages in the genome that can give us some instability. It has a type of glycolysis that is not the typical glycolysis. has an alternative type of glycolysis that is called the Edner-Dodorov pathway. It's an obligate aerobe, so that means that it grows only in the presence of oxygen. And as I said, because of the name Pseudomonas, it has a little bad reputation, even though, as I said, you can drink it and still you will survive. So it's not a bad bacterium there. So here comes our mission, and is to start with something with some plus and minuses and do something that is much, much better than nature has produced by itself. So then we have to run into this kind of pipeline where you start with something that is interesting, may have interesting properties, is naturally occurring, is not natural, but it's quite unpredictable, and it will not obey our orders. So eventually what we would like is to have something like that, as I said before, that something is completely engineered and therefore that is entirely, able to respond to our commands. So between that and domestication, we may entertain another stage that we can call cyborgization. And I don't know whether you heard this word before, but cyborgization is about combining existing traits with artificial traits. So what is involved in cyborgization? Well, what is involved is the enhancement of innate desirable traits, the replacement of some traits by better ones, if you happen to have them, to knock in entirely new traits and elimination of drawbacks. So cyborgization is about combining things that are completely artificial with things that are already there. So in the case of P. putida, of Pseudomonas putida, this is something that you can do by taking a look to the genome and then deciding what genes you want to keep and what genes you want to eliminate. Up front, you start by thinking what genes you want to eliminate. And the work of someone called Esteban in my laboratory has for various years trying to identify, to go through all the genome of Pseudomonas putida and then eliminate some of the proteins, some of the genes that we thought were not particularly interesting. Which ones were not particularly interesting? Well, for some applications, it happens that the natural program of Pseudomonas putida is not the same that the program that we would like it to run under our commands. For instance, bacteria have flagella to move around in the environment. Whether you want a flagella in a bioreactor, for instance, that's something that, the flagella is our enzyme that will consume energy and NADH and so on, and it's not interesting to have a flagella in bacteria for this type of applications. Also, it may form a large lipopolysaccharide that is, again, a waste of metabolic resources, and we don't need it in our settings. And for instance, it has also some prophages that are there like parasites. So by making this type of examination of the genome, you can identify what parts of the genome you want to get rid of, and then at the end, you may have a bacterium that is, say, more predictable because you have eliminated some features that you don't like to have. So this is what we did. We eliminated big chunks of the genome of Pseudomonas putida, involving sulfurase antigens, things that were not desirable, some antibiotic-resistant genes, some transposases. I mean, we made at the end a bacterium that was performing much better than the natural isolate. So, for instance, just by deleting the flagella, you get bacteria that are way more tolerant, way more resistant to oxidative stress. So paraquat is a trigger of oxidative stress in bacteria. So this is the survival of the cells in the wild type cells, and this is the survival with the bacterium that is deleted of these flagellar genes. Okay, so you see that you have at least one order of magnitude more resistant. And this is another diamide, is a compound that depletes the cells from NADH. And then you see here that, again, the survival and the capacity to live under oxidative conditions is much better in the strain that is deleted of the flagella. And well, you can really see that by making this type of chromosomal addition, of genomic addition, at the end you have some variants that, for instance, have more ratio of NADH, the reduced versus the non-reduced, the oxidized version of these compounds here, so you can really get a better, say, performance in redox conditions. And also, for instance, the strains that are deleted of the flagella do better in surviving a stationary phase. So by making these little modifications, you indeed enter the possibility to, improve the biotechnological performance of an otherwise natural bacterium. But there are other traits that you don't want to have in a chassis, in a bacterium that you want to really program and domesticate. And one of the worst is the formation of biofilms. You know that bacteria and all types of microorganisms like to be stuck to surfaces. And they form these structures that are called biofilms, where basically bacteria colonize the surface. They secrete some compounds that act as a glue for holding the bacteria close to each other, and then they colonize surfaces. So this is not nice. I'm not sure that you have seen biofilm many places in your bathroom, probably. I mean, in the shower or something, you always find these little sticky, mushy things on the walls that are bacterial biofilms. So you don't want to want biofilms. Instead, you want to have something completely different. You want to have something like this. So this is how a biofilm could look like. You kind of association, a messy association of different bacteria. But as a synthetic biologist, as an engineer, I would like to have something more similar to this. So I would like to have a system to program bacteria to stick to surfaces, but not in a messy fashion, but in a controlled fashion. And also, I would like to have a system through which bacteria interact to each other, not randomly, but following a given pattern that I can engineer by means of artificial adhesins, okay? So I would like to go from here, this messy biofilm, into here, okay? So how can we go from here to here? Okay, so here comes again the power of synthetic biology. And one of the things that you can do is to go through the genome of this bacterium, identify the genes that encode outer membrane associated structures like fimbriae, LPS, flagella that I mentioned before, and then end up with what we have called the naked variant of Pseudomonas putida, okay? So in this naked variant, you keep all the biochemistry, all the nice properties of Pseudomonas putida, but you delete, you eliminate, you erase from this bacterium everything that you don't want to have. So we did that, and here's the what we call the surface naked Pseudomonas putida, that, you know, even just by looking at it, it looks like more friendly. You know, it looks like a friendly bacterium. It looks fat, happy, and very, very well behaved. And also, as we deleted some antibiotic resistances, then the strain is more sensitive to some antibiotics, and therefore is more amenable to genetic manipulations. So what is the advantage of having a bacterium that is naked? Okay? Okay? This is the trick. That if you have something that you undress, then you can redress it with different things, okay? So obviously you have a wild-type bacterium without all these pili and all these things, then it's difficult to do anything with them. But if you make the surface naked or semi-naked, then you can now take this puppet and put other things, and then every time that you put a different thing, you have a different puppet, and you have new possibilities of doing some interesting applications. So what things you can put on the surface of this naked bacterium? Well, you can think on many, many possibilities, and I will just mention a few of them, not all of them, but some of them. One of them that we have done, this guy in my laboratory called Pavel Borsak, a Czech postdoc, what he did was the following. You know that there are bacteria out there that are able to degrade cellulose. Cellulose of paper, cellulose of different origins, straw and these things. And it happens because they have on their surface a multi-protein complex called cellulosomes. So cellulosomes are super complex, protein, assemblies, that are actually like actual cellulose grinding machines. So, but it happens that these type of structures only occur in some bacteria that are way far from Pseudomonas putida and are difficult to handle. In some cases are extremophiles, and well, they are not very useful as elements for cellulose biodegradation in vivo. So what Pavel did was to engineer on the surface of Pseudomonas putida a display system that enabled the assembly of some components of these cellulosomes on the surface of the cells in such a way that now the cell acts as a scaffold for a multiprotein complex that act on the outside to degrade a polymer, okay? And this is something that I consider to be nice because, you know, once you have this polymer degraded, then you produce glucose, and then the glucose can be entered into this cell and can be converted into other interesting compounds. But you can think also to put other interesting activities on the surface of a naked bacterium. For instance, one thing that we are trying, we are not there yet, but we are trying, is to see whether Pseudomonas putida can be used as an agent to capture humidity in dry soils in the night. You know that there are some lizards in deserts that never ever drink, and what they do is that they have in the skin they have some spines that are coated with a protein that is able to capture small amounts of water. And even though these guys never drink, they capture enough humidity, enough water to go on. Then, what project that we have in the laboratory is to see whether we can genetically encode these proteins in the genome of Pseudomonas putida and then display these proteins on the surface of Pseudomonas putida in such a way that now these cells can act as attractors, as suckers of environmental water in the night, and perhaps to change the ecology of some micro niches that are present in drylands. And theoretical ecology will tell you that just a marginal change in the amount of water that you have in soil will have tremendous consequences in terms of the ecology of the place. So that's something that is quite nice. Okay, so this opens a bit the idea, and I leave you this as an aperitif because we will have a special class about that, we can engineer bacteria through the tools of synthetic biology, not only for industrial applications, but also for environmental applications involving the release and the inoculation of the target sites with the bacteria and the synthetic biology agents that we construct in the laboratory. This is just one example of how some bacteria that were developed in the laboratory were used for recovering of some corals in Australia. So if you are curious, you can take a look to it. And so the idea is to have some modifications here and then re-inoculate them in a coral, and then the experiments show that when you do that, at the end, the system recovers its activity. But we can put other things on the surface. And if you remember yesterday's talk, I show you a bit about camel antibodies and all the benefits that you have of a camel antibody, because as I mentioned yesterday, they have as part of their structure a small domain that is called the VHH domain that can be separately produced. You can split it genetically from the rest of the molecule, and this domain has the capacity to bind strongly to an antigen that the camel was exposed to formerly. So over the years, we have developed a large number of tools that enable to display functionally camel antibodies, or this small part of the camel antibodies, on the surface of different bacteria, E. coli and Pseudomonas itself, in such a way that now the entire surface of the cell becomes coated with these antibodies, and therefore now the whole cell has the affinity to stick to target molecules, whether they are displayed on another cell or they are displayed on a surface. And this is something that, technically has been solved, and now we have the possibility to use antibodies as artificial adhesins. And artificial adhesins will will serve two purposes. One of them to use them for sticking bacteria to each other, but also to target the adhesion of bacteria to specific surfaces, as you will see in a moment. For doing that, you need to have some theoretical frame that will predict what will be the consequences of having adhesins distributed on the whole surface of the bacteria. They can be in the poles, they can be in the body. And then depending on where you have the adhesin, the type of three-dimensional structure that you obtain where you mix bacteria that express on the surface an adhesin, and the one that displays on the surface, the target for the adhesin, can be completely different. You can have pole-to-pole interactions, you can have body-body interactions, you can have pole-body interactions. And this is something that can be modeled with the tools of systems biology and say Monte Carlo simulations, and you have a whole bunch of technologies that enables you to make a prediction of what happens when in vivo you put a given adhesin in a specific part of the cell, you shake them up, and then you ask the system to predict what type of self-assembly you will have of that bacterial consortium. Okay? And well, this is something that someone in my laboratory called David has been doing, and you can play with different, on the one hand, on different constructs that you make in the laboratory, and also with different colors, just to validate the concept. So for instance, you can have bacteria that are tagged with a gene fluorescent protein that display on the surface an antibody, and then you can have other bacteria that display the target, the antigen that is recognized by the antibody with a different color, and then you mix them up and see what happens. You can make some theoretical predictions on the shape of the flux that you will get when you make these mixtures, or you can make actual experiments, and then you can match the type of say clusters, the type of flux that you get theoretically with the type of flux that you get in practical terms. And in general, by doing that, you can construct, say, a consortia with a specific three-dimensional architecture, okay, where bacterium A touches bacterium B by following a given rule that you can model and you can quantify. Okay, I think it's time for a little coffee break, and I will return later with just a few slides to see some other, to share with you some other features that you can engineer in a chassis, in this case, Pseudomonas putida. Once you have the genome streamlined, you have deleted all the bad parts that you don't like, and you have a good chassis, a good standardized chassis that you can really use to go into the environment and into industrial applications, okay? But let's pick that up after the coffee break. So let's be back by a few minutes before 4:11, okay? Okay, see you in a little while.